Imagine you're a diligent grad student who spent weeks poring over a breakthrough paper, only to watch your carefully replicated experiments fizzle into inconsistency, and you stumble upon the Replication Crisis Paradox, a narrative woven from true stories of landmark studies in psychology, cancer research, and economics that refuse to stand up under identical tests, revealing this isn't a fluke but a deep challenge in how we do science. What if rigorous peer review and p-values alone aren't enough? The paradox goes beyond bad actors or sloppy labs. It highlights subtle forces. Tiny shifts in experimental wording derailed the power pose effect when 20 labs tried it. Mouse strain differences erased promising cancer drug results in biotechs. Billion-dollar failures, cultural nuances flipped, famed economic games on tax honesty when run in different countries. And each example shows how researcher degrees of freedom, minute procedural choices, and context can turn a real effect into a phantom. Picture a junior psychologist under publish or perish stress. The miracle finding in your first paper vanishes when you run it again with new participants. What now? LSCC for science. Sometimes called open science, teaches you to pre-circle your design, locking in hypotheses and analysis plans before data collection, share raw data and code for full transparency, and pursue multi-site collaborations so that no single lab's quirks can sway your results. At the heart of tackling the crisis lies the question, how do we turn shaky findings into robust truth? The answer must be systematic, embrace larger sample sizes, like genome projects combining thousands of patients to weed out false leads, adopt registered reports to peer review methods before results exist, and cultivate a culture where null results are valued, preventing the toxic file drawer effect that buries failed replications. To sail past this paradox, scientists cultivate 6 key practices: the curiosity to question every result, even your own, the precision to document every protocol step so another lab can follow it exactly, rigorous planning from pilot tests to full-scale studies, meticulous record-keeping of data and code, collaborative openness, inviting others to challenge and verify your work, and resilience to publish and learn from failed replications, seeing them as progress rather than embarrassment. Worried that transparency means extra work? Yes, these practices demand time and social courage to share unfinished results and unexpected nulls. But they also teach preemptive moves. Set up data management systems like CERN's collaborative platforms, schedule regular reproducibility audits among lab members, and follow biochemist John Ioannidis' rule of reproducibility checks at every milestone, turning potential breakdowns into pillars of trust. Ready to rewrite the story of your research? Begin by precepting your next study. Share yesterday's data set on an open repository. Draft a replication plan with 2 partner labs. Pick one open science practice like logging every change in your code, and dive into 1 case study a week, asking, what step did they miss and how can I do it better? In this evolving saga, your scientific life, you'll transform from chasing elusive p-values into building discoveries that endure under the toughest scrutiny. The replication crisis refers to the widespread observation that a substantial fraction of published scientific findings cannot be reproduced when identical methods are applied. Despite rigorous peer review and statistical thresholds, many studies yield results that vanish in follow-up experiments. This paradox undermines confidence in scientific literature and raises questions about research practices, statistical inference, and publication norms. Understanding its root causes is essential for restoring reliability across disciplines. In psychology, a large-scale effort reproduced only 39 of 100 landmark studies using the same materials and protocols. It's like following a trusted recipe exactly, yet ending up with a different dish every time, even though each step was executed faithfully. Which, by the way, is true. Reproducibility means obtaining the same results from the same data and code, while replicability means observing similar outcomes in new data collected under the same protocols. Both are pillars of scientific validity, but address different stages: computational transparency and experimental robustness. A failure in either dimension signals potential flaws in methodology, reporting, or theory. Distinguishing the two helps target specific remedies. Better code sharing or more rigorous experimental design Genomics papers often share raw sequencing files, reproducibility, yet attempts to replicate biomarkers in independent cohorts frequently fail. Reproducibility is like reprinting a photograph from the same negative, whereas replicability is like taking a new photograph under identical lighting and expecting the same scene to appear. Researchers often explore multiple analyses and report only those yielding p-values below 0.05, a practice called p-hacking. Selectively choosing covariates, outcome definitions, or time points inflates false positive rates. Questionable practices, such as harking, hypothesizing after results, further bias findings away from what a truly impartial test would show. These micro-scale behaviors accumulate to produce published results that reflect analytic flexibility rather than genuine effects. A biomedical study tests 10 different endpoints, but highlights only the single significant one, leading others to fail replication when testing the originally unreported endpoints. It's like rolling dice 10 times, but only telling your friend about the one roll that shows a 6, then claiming the die is biased. Journals prefer novel, positive findings over negative or confirmatory studies, creating a file drawer effect where null results remain unpublished. Academic incentives, grants, promotions, prestige, reward striking discoveries, not careful verification. Consequently, the literature becomes skewed toward false positives that cannot withstand replication efforts. Addressing this systemic bias requires altering incentive models to value rigor and transparency. Meta-analyses often reveal a skewed distribution of effect sizes, with small studies showing exaggerated effects that later vanish in larger trials. It's like a news channel that only reports storms and earthquakes, making viewers think disasters happen daily, while days of calm go unmentioned. Many studies are underpowered, using sample sizes too small to reliably detect the true effect sizes they investigate. Low power increases both false negatives and the probability that any significant result is a false positive. Underpowered experiments also yield effect size estimates that are wildly variable, making replication unlikely. Ensuring adequate power through pre-study sample size calculations is fundamental to reproducible science. A social science experiment with 20 participants per group reports a large effect, but when repeated with 200 participants, the effect shrinks or disappears. It is like trying to measure small temperature changes with a thermometer that only reads tens of degrees. Tiny fluctuations are simply lost in the instrument's noise. Real-world phenomena often depend on myriad contextual factors, environment, timing, individual differences that are difficult to control or record fully. Even identical experimental conditions can mask subtle variations that critically influence outcomes. In biology, cell culture reagents, lab ambient temperature, and animal microbiomes can all sway results. Acknowledging and systematically documenting context is crucial for true replication. A mouse behavior study replicates only in labs with matching ambient noise levels and cage enrichment protocols. It is like baking bread. The same recipe fails unless you match the local humidity, oven calibration, and flour mill, all subtle but decisive variables. Poorly specified theories yield ambiguous predictions, leaving researchers to operationalize constructs in ad hoc ways. Divergent operational definitions across labs produce results that test different underlying phenomena. Without clear theoretical anchors and standardized measures, replication efforts may inadvertently probe different constructs. Strengthening theory-me-measurement links enhances both the interpretability and replicability of findings. Stress is measured via self-report, cortisol levels, or heart rate variability in different studies, yielding inconsistent replication. It is like different teams measuring vehicle speed with odometer readings, radar guns, or driver self-report. Each captures a different facet and may not align. Registered reports require study protocols and analysis plans to be peer-reviewed before data collection, committing authors to predefined methods and reducing p-hacking. Open data and open code mandates allow independent reproduction of computational analyses, incentivizing publication of null results, and replication studies counteract publication bias. Together, these reforms aim to realign incentives toward reliability instead of novelty. Journals adopting registered reports have seen null findings published at rates matching positive findings, improving the literature's balance. It is like building a house to inspected blueprints. Once approved, builders must follow the plans exactly, ensuring structural integrity. Overcoming the replication paradox demands a cultural shift valuing transparency, collaboration, and humility about uncertainty. Training early career researchers in robust statistical methods, pre-registration, and data sharing normalizes these practices. Funders and institutions can reward replication work and rigorous negative findings, ultimately fostering a collective ethos of verification rather than hyper-competition will stabilize the edifice of scientific knowledge. Multi-laboratory consortia pooling resources to systematically replicate high-impact studies across different contexts exemplify this approach. It is like an open-source software community where many contributors review and test each module, ensuring the final program is robust and bug-free.